parts, the basic structure. We're going to talk more about the parts of viruses. Last time we talked, let me get these up here. We talked about different kinds of shapes. Questions about any of that? So let's see here. When we ended last time, we were talking about nucleic acids and the variety of formats that the nucleic acids could be in. Why do we have a shadow on there? They could be DNA, single-stranded or double-stranded. They could be RNA, single-stranded, positive or negative or double-stranded. Some viruses are actually diploid. They might have 2 copies of their genome. There's all kinds of different variabilities about what their nucleic acids can be. And one of the things I think is most interesting about viruses is what it takes for them to replicate all of these weird genomes. And so we're going to talk about that when we talk about how the viruses replicate. We're going to first talk a little bit more about the parts of viruses. And we can't cover every single detail, so we're talking about generalities in terms of parts. And then later when we talk about diseases caused by viruses, we'll go into more detail about some of them that are especially important for causing disease. So in terms of parts, the protomers, those are the proteins that make up the capsid. The nucleic acid is the genome. If you put those together, when they're associated with each other, where'd my pointer go? It's called the nucleocapsid. So the capsid associated with the nucleic acid together is called the nucleocapsid. And in some cases, that's pretty much all there is to the virus. Some of them have more parts. But if you're talking about the nucleocapsid and the parts that are also associated with it, the nucleocapsid is just the protein around the genome. So in terms of optional features, some of the viruses have what are called envelopes. Whoops, I almost spelled that wrong. Not all virions, not all viruses have envelopes of them. Those that do get them from the host membrane. We talked about this just really briefly the other day. And Hannah, you asked something useful and interesting about that. What was the deal?
Is that how they hide from the immune system?
Right, is that how they hide from the immune system? So the envelope itself is just made out of the phospholipid bilayer from the host. But as they form, the important features of the envelopes that are associated with them are the proteins that are stuck into them. And this figure shows an envelope virus. In this cartoon, this yellow thing going around here is the envelope. And they can have proteins associated with the envelope, and those are known as spike proteins. So among the proteins that can be associated with the virus, one of the important ones are the spikes. Some books and some virologists refer to the spikes as peplomers. So you might see that word too. I always call them spikes. They're important for a few different things. In this picture, they're shown kind of sticking out a little ways. And one of the important things that they are involved in is related to Hannah's question, and that was the fact that they are what are antigenic. They're the antigenic part of the virus. So when a person is exposed to a virus and then make an immune response against them, against the viruses, this is the main thing that they first experience, that our immune system see as foreign. So imagine if you inhale a virus. What's exposed on the virus is the phospholipid bilayer, which is not particularly interesting, and the proteins that are associated with the surface of the virus. So these are the antigenic parts. The part that we make antibodies against. So antibody responses to this. That's one important thing about them. Another thing is that this is part of what is used for classification of viruses. When you guys, we'll talk more about this later when we talk about influenza, but you guys have probably all heard about H1N1 and H3N7 and all the different H's and N's. Those are the numbers associated with them, like H1N1. The ones refer to different types of the spike proteins on their surfaces. And so when we categorize them, we base it on the type of spike that's on the surface, because that is what the antigenic response is to, and that's one of the ways that we can describe them. So this is the basis for identification of strains. There's a whole bunch of different H's and a whole bunch of different N's, and we'll talk about that when we talk about influenza specifically. The other really important feature in terms of the virus, so these are sort of all about us, right? The first two are human interest things in terms of how we use them and think about them. From the virus's perspective, the most important thing is that these are involved in attachment. So when a virus associates with the host, and we're going to talk about what it takes for the virus to replicate itself in a little bit, the first thing it has to do is stick to the host. And in the envelope viruses, and even in the, well, we'll talk about those later. But the important thing that they have to do is associate with the host, and it's very specific in terms of how it does that. The spikes are responsible for which kinds of host cells can be infected by the virus. So when you're talking about H1N1 and H2 and H3 and all those different things, those different H's are specific for different species of animals. So you probably heard that influenza can infect pigs and birds and bats and humans. That is based on different spike proteins and their affinity for receptors on the surfaces of our cells. So this falls into the category. Remember on the first day when we talked about the different core concepts in biology? Put this into your framework of core concepts. This is a case where structure and function are really important for a particular molecule and what it does for the virus. So the structure of the spike proteins is responsible for its ability to stick to particular kinds of cells of different animals, and also in terms of different tissues in the body. So for influenza virus, the spike proteins are specific for your respiratory epithelium. How about for Zika virus? Where do you suppose Zika, based on what you know about Zika virus, what do you suppose it sticks to? You guys have heard, Zika's not in the news anymore because it's not affecting us, it's affecting other people. So we just don't care that much. Sad but true. So what do you know about Zika virus, Ben?
Is it selected to reproductive cells?
It has to get into that, well, at some point, since you know it's sexually transmitted, it has to somehow get associated with those cells as well. But I was thinking more in terms of the kinds of diseases it causes. What's the main disease associated or condition associated? Small head, is that what you said? Yep, microcephaly. And people, when Zika first came out in the news, people didn't really know for sure that that was what was causing the microcephaly and the small head development. But what they found out now is that the spike protein on Zika virus binds specifically to receptors that are on the developing neurons in unborn babies, in the glial cells. And so the glial cells have proteins on their surfaces that are the targets for the spikes on the surfaces of Zika virus. And that's how come Zika virus infects those particular cells and why it causes that particular birth defect. Same thing is true of polio. For polio virus, which we'll talk about another time, it has spikes on its surfaces that are specific for particular cells in the digestive tract. So if you got really mild polio, it wouldn't be paralyzing. But also some of the cells of your nervous system that are responsible for muscle movement have the appropriate receptor on them for the polio spikes to bind to. And so that's what controls what kinds of cells. That's what this means. Responsible for tissue tropism. This is what controls what cells are infected. So the reason it controls that is because it has particular receptors. So that is not what controls it alone. It's along with the host cell receptors. So in terms of in terms of the parts of the viruses that are really important for causing disease, this is really huge because it controls whether we get infected by the virus or not and what kinds of cells it binds to. Let's see here. So these are proteins in the capsid. Spikes are associated with the capsid. Spikes are big. There are some enzymes in there. We'll talk about in detail when we talk about replication of the viruses. But based on, you can make some predictions, based on what we said about the different genomes of the viruses, you know there's got to be some enzymes in there that are responsible for replicating the viral genome, right? Because if you have a virus with an RNA genome, how on earth would we ever replicate an RNA genome? Because our cells are going to be responsible for being the factories. So oftentimes the enzymes are involved in replication of their nucleic acids.
Are you talking about host enzymes or viral enzymes?
We're talking about viral enzymes. Viral enzymes in the capsid. This is nucleic acid replication. So an example of one that you probably heard of before is reverse transcriptase. And there are other enzymes. Some of them are important for processing. We'll talk about them as we go, especially when we get to the section on HIV. Okay, parts of viruses. Let's talk about what does it take to reproduce the viruses. Does anybody have any questions about, here's a vocabulary word to mention. This figure shows a couple different viruses. This one is the rabies virus. It's got a characteristic bullet shape. It's always shaped like that. This is a herpes virus. And notice how it's all wobbly and irregular around there. Did you guys learn the word pleomorphic in lab yesterday? You talked about, no? Pleomorphic refers to variability in shape. So if a cell is pleomorphic, it means it can have variability in the shape of the cell. The same thing is true of virions. Virions can be also pleomorphic, having different shapes. So this one is odd and irregular because it's a pleomorphic virus. This one is uniform and predictable, and it's called a bullet-shaped virus. And the reason for that shape is that in this case, the rabies virus has its envelope attached tightly to the nucleocapsid. So these are false colored. They're not really that color, right? But the blue and gold stuff here in the middle is the nucleocapsid. In this case, the envelope is tightly attached to that. In this case, the nucleocapsid is this kind of pinkish red thing. And it's got tegument proteins inside just under the membrane, or under the envelope, but the envelope is not tightly associated with this. You'll hear that word pleomorphic at other places too. Okay, so let's talk about reproduction of animal viruses. So reproduction is kind of an unfortunate word for this. There's some stuff in the book, in these chapters, about reproduction of bacterial viruses, a bacteriophage. We're not going to talk about that right now. We're going to talk about bacterial viruses in the context of exchange of genetic information. So it's going to go in with the genetic stuff. And then we'll go back and review it because in terms of human interest in viruses, in bacteriophage, they fit more into ecology and into genetics than they do just by themselves. So reproduction, I would say that's kind of an unfortunate word because we say viruses can't reproduce themselves, but they get reproduced by the host. And so this figure is an overview. We're going to go into some detail that shows what's involved in viral reproduction. First, the virus particle, or the virion, sticks to the host cell, and like we said, that's if it has spike proteins, the spike proteins will be involved. In some cases, the virus has affinity for the host cell, but it's not really catalyzed by a spike protein. It has to get into the cell, and there's a few ways that can happen. Then it gets uncoated, and the genome, the viral nucleic acid, gets into the cell, sometimes in the cytoplasm and sometimes in the nucleus. I think we're going to just talk about it in the nucleus today. then the virions get assembled and they get out of the cell and go on to spread within the host or within other hosts. So the first part of that is what the book calls attachment. Sometimes different books call it absorption. Attachment or adsorption. Not absorb like a sponge, but adsorb. Adsorption. So how does the virion get to the host cell? Initial collision between the virus and the host cell is random. It's not like it can swim up to it, like a bacterial cell can, swim through the water and be in our water supply, or swim around in our bodies to find the right place. It's an initial random collision, but then for it to be a productive interaction and for the virus to reproduce, there has to be a specific protein or glycoprotein on the virus that matches a particular receptor on the host cell. So this is a specific step. We've already talked about that today. Specific for particular receptors and viral proteins. And we'll talk more about that when we talk about particular diseases like HIV and influenza. So the next thing that has to happen is that it penetrates into the host cell and it uncoats, penetration and uncoating. So that is, in this figure, referred to as entry into the host, entry of the viral nucleocapsid into the host. So that can happen a couple of different ways, depending on what virus it is. So, oops. So this shows entry when the virus has an envelope around the outside. So remember, the envelope is just made out of the plasma membrane from the cell it just came out of. So the spikes on the surface of the cell bind to the receptors on here, and what can happen in this case is that the envelope fuses with the plasma membrane of the host cell. Just like two soap bubbles coming together, and two little soap bubbles make one big soap bubble, the same kind of thing happens with the membrane. And then the nucleocapsid that has the genome gets dumped into the cytoplasm of the cell. When that happens, some more stuff has to happen to get that out of there. We'll talk about later. Ben?
So when the virus first begins, will it not have an envelope until it enters the host cell?
It doesn't have the envelope until it leaves the host cell that made it. Because the virus, we'll talk about this later, but what happens is as it leaves the cell, those that have an envelope get processed and then bud off of the cell. And at that point, they get their envelope from the cell that they're leaving. Which means that the cell has to be intact as it leaves. If you have cells that or viruses that have envelopes on them, they can't just break the cell open and kill it wildly, because then they wouldn't have an intact membrane for them to escape from. So this is one way that this can happen, by fusing. Other way is by endocytosis. So you guys probably learned about receptor-mediated endocytosis in cell biology, right? The clathrin-coated pits and the receptors that trigger the rearrangement of the cytoskeleton. Does that sound familiar? So that's what's going on here. The virus binds to these receptors, and that triggers endocytosis. And here, it looks like the virus has its own envelope, and it's got an additional layer because it's in an endosome. And in that case, what happens is it goes through the endosomal fusion process, and you get the acidification of the endosome. Remember that from cell biology? There's ATP pumps in the membrane, and it pumps protons in there at the expense of ATP, and it acidifies this vesicle, which results in the encoding of this. A couple different ways it can get in. So those are with envelope viruses. With non-envelope viruses, it can also go in by endocytosis, but you don't have that second membrane. So the thing gets taken up, and in this case, the nucleic acid gets out of that endosome and into the cytoplasm to go where it's going to be going for replication. So I'm going to write that down. Fusion with host as a note to yourselves. That's with envelope viruses. Or endocytosis. So that could be with envelope viruses or non-envelope viruses, right? You can't fuse with the host cell membrane if it doesn't have its own membrane to be fusing with. Christian?
Do the glycoprotein spikes, do they mimic something that's generally beneficial to the cell? Or why else does the cell want to take?
That is a good question. So what happens, and this has to do with selection and evolution. So they don't necessarily mimic something because there's no, from the point of view of the cells, there's no kind of cell that you want to have invaded by a virus, right? So what has to happen is that as the virus spikes evolve, they have to be, there's selection for those new forms that can still bind to a host cell receptor. So if they change too much, then they can't have a productive infection of a new cell. In terms of, was your question whether the virus spike mimics anything? Because that would be really good from the point of view of the virus, because then we wouldn't have an immune response against it. If you had a spike protein that looked like something else, you wouldn't raise antibodies against it.
And cells would readily uptake it.
And the cells would get tricked into taking it up. I can't think of any examples of that. That doesn't mean there aren't any, but that's a good question. Any other questions about anything so far? Penetration, coding. Okay, so here's the part that I suggested that you guys think about over the weekend, and that is replication of the viral genome. And remember, I said this is where we're going to go from one chapter to another, so don't get shocked as I turn the page here. This is in chapter 27. So chapter 27 has a section about replication of the viral genomes. And I put those figure numbers are on Moodle or everything, so you guys can totally find this. We're going to look at a couple of examples of how viral genomes get replicated. I'm not going to talk about every single one, but we're going to talk about those that are either important for health or ones that are especially important for biology because they represent variability in terms of what happens in the world. So the one that is the easiest to think about, I think, is replication of double-stranded DNA viruses. Double-stranded DNA viral genomes. And the example, this is figure 2717 from the book, and the example they're showing us is the herpes virus family. Whoops. Herpes viruses. So this includes not just cold sore herpes and genital herpes, it's the family of viruses, which also includes the chickenpox virus, varicella zoster virus, and we'll talk about that some more when we talk about diseases. What they all have in common is that they have a double-stranded viral genome. And in order for that to replicate, here's what happens. So this is, again, figure 2717. It shows adsorption here. The virus has to stick. This is another example of an envelope virus with spikes, so it has to stick just to the right kinds of cells. And there are receptors for the herpes viruses on a lot of different kinds of cells in your body, but what do you suppose are the ones that first come to mind? This causes herpes, cold sores, chickenpox. Epithelial cells, yep. So epithelial cells have the right receptors. Also some of the do. So the virus binds on here, it gets in, you can see there's fusion here, right? That's what's being shown. And then the nucleocapsid binds its way to the nucleus. So this is where your cell biology comes into play. It doesn't just float through the cytoplasm and bump into the nucleus. It actually gets grabbed a hold of the cytoskeleton. And there's, remember what moves things along the cytoskeleton, along the microtubules? Dynin grabs a hold of the nucleocapsid and associates it with the microtubules and walks it on down to the nucleus. And then the nucleocapsid associates with a pore. You learned about the nuclear pores, right? They're cool basket-shaped things. It binds there, and then it gets exported into the nucleus. So the DNA genome is getting replicated in the nucleus. That totally makes sense, right? But what's happening is a little bit different. It forms this circularized form of the DNA. So first thing is circularization of the genome and transcription of the immediate early genes. So this gets circularized like that. There's classes of genes in the viral genome called the immediate early genes, the early genes, and the late genes. Or in this case, the proteins, because they're pointing to the proteins. Those things that have to be done immediately include things that are required for replication of the genome. So this is a virus that uses its own DNA polymerase. So you've heard of DNA polymerase before, but it's not using the host cell's DNA polymerase. It uses viral DNA polymerase. It brings its own gene for a viral polymerase in. And what's happening here? So the immediate early genes get made. They're shown being transcribed here. It goes out into the cytoplasm to be translated, and then they come back in. And the first one they're showing here is immediate early proteins that stimulate genes, production of genes that are needed early on in the cell. Some of the ones that are called early, the early proteins include those, including the DNA polymerase that I just mentioned. And what happens with this particular genome is it makes the circularized genome, and then the DNA polymerase goes around the circle, making multiple copies, which are called concatameric DNA, yielding concatamers. Multiple copies all connected together, which then have to be processed. So that would be the early proteins. The late proteins, those that get transcribed at the end, include those that are needed for virion assembly. So the virion, the capsid it's going to form, is made by the late genes. So that's the last thing that has to happen. So late proteins get made, they get moved into the nucleus where this DNA gets processed and chopped into individual subunits and then put inside of the capsid. So in this case, there's actually, so remember we talked about self-assembly of the capsid last time? What was the deal with that? How could the capsid assemble on its own? Remember there's affinity, so in this one it shows an icosahedral, so there's triangular shaped monomers, right? And they all come together and they have affinity to make this dome shaped thing. It would be just too amazing if that all crystallized around the genome, right? That would be super unlikely to have that wrap around. They make the structure and then they have an enzyme that someone cutely named the PACase or something. I don't remember it because it's just too cute. But there's an enzyme system that associates with the capsid and grabs a hold of the DNA and then pumps it in. And it does that at the expense of ATP. So it's transporting DNA into the package, and it gets in there really tight. And one of the things that virologists used to study for a long time was how on earth can the DNA get in there so tight, because they have a size limit for how much they can fit in there, and it's packed in there pretty tight. Okay, so what happens? This is going on in the nucleus. And in this case, we said it was an envelope virus. So in order for it to get that envelope, it has to be processed and sent out through the cell, and that's going on here. It buds off of the nucleus, gets into the Golgi. And how about this? All of these proteins along the outside. It doesn't tell us a lot about what happened there, but what's missing between the nucleus and the Golgi for an envelope virus in terms of the spike proteins? It doesn't show how the spike proteins get there, but you know, since they're membrane bound, what's going to happen with the spike proteins is they're going to be translated on the rough ER, right? They're membrane bound proteins that are getting made on the rough ER, just like our membrane bound proteins. They're going to get expressed out here. Here they are. This comes through here, and it's going to leave the cell by exocytosis with all of those proteins made on the surface. So this kind of goes back to some figures in chapter 6. We'll look at those again later when we talk about this. So what are the highlights in here? There's genes that are expressed at different times to make this work. That's one thing. There's the immediate early, the late. They have to be expressed at the correct time so that this all works right. So you have the genome made and all ready to go when the capsid gets made. What else is important here? The fact that the virus brings in its own DNA polymerase. So why do you suppose it wouldn't work for the virus to use the host cell's DNA polymerase? Ben?
Because we don't have circular DNA.
That's a big one, yeah. We don't have circular DNA, so we have a different kind of polymerase. What would be another thing in terms of our cells and their DNA polymerase? Like the timing in the cell cycle. This virus doesn't know what kind of a cell it's going to infect in terms of where it is in the cell cycle. It just knows that it has the right receptor, and it doesn't really know. We always anthropomorphize things, but it doesn't really know anything. It just binds to the receptor because it has affinity for it. So it binds to the cell, it gets in there, but the cell doesn't really necessarily have its DNA polymerase activated and replicating DNA at all times, right? That's something that happens only in a certain part of the cell cycle. So if it just binds to an epithelial cell and gets in there, there's no guarantee DNA polymerase from the host is going to be active. It has to bring its own and express it, transcribe the genes for it, and translate the proteins for it when it comes in. Otherwise, there may be no polymerase in there. Other highlights about that. Do you guys have any questions about DNA? So this is conceptually straightforward because DNA gets replicated, genes get transcribed, proteins get made, just like happens in our cells with some variations that have to happen in order for it to work in viruses. So, C figure. I know you're going to have to read about it. You guys, I hope you guys know you're going to have to read about this and think about it some more, right? See this figure. Okay, 2717, that says. All right, let's talk about the RNA viruses, at least start. So what about this is replication of RNA viruses. So there's more options with RNA viruses and more things that are conceptually different to us. If you have, so remember there's positive single-stranded RNA, there's a negative single-stranded RNA, double-stranded. So we talked about this last time. What's the difference between those two? Tony? Right, this can be used as mRNA, yep. In some cases it's also the genome. So it's not limited to being mRNA, but it's the kind that's used as mRNA. So then what would the negative be? The negative is the complement. Is that what you're going to say, Ben?
Yeah.
It's the complement to the mRNA or to the positive strand. And if you have a genome that has either of those two and is single-stranded, in order to reproduce your genome, what is the rule about making nucleic acid polymers? How do cells do it in general?
Go through like a double-stranded form.
Yep, they have to go through double-stranded form, and that means that they have to have a template that they're going to build the complement on. And so in both of those cases, both the positive strand and the negative strand, in order for them to replicate, they have to have a template that's complementary to the genome that they're making. So if your genome is positive single-stranded RNA, that's the genome, that's what you use as mRNA. You still have to make the negative strand in order to reproduce more positive, because the goal of the virus is not to make us sick or anything. The goal is just to make more virus, right? So if you're going to make more of this, to make multiple copies of the single-stranded RNA, you have to make the complement first and use that as a template to make a whole bunch more. Okay, so let's look at what that looks like. Here's a figure from your book. This is the positive, this plus minus means double-stranded RNA. This one has both strands. So we can use the positive strand to make protein, use the positive strand as a template to make the negative strand, which is then the genome. This squiggly red line represents the protein before it gets all folded up, and it makes the capsid proteins, and this goes in there, and that's nucleocapsid. That's, double-stranded is conceptually familiar to us. Here's the positive genome. The positive genome can be used to make protein. It can be used to make negative-stranded RNA. And the only purpose for doing that is to have a template to make more of this, right? So that would have been helpful for the book if they would have shown many, many copies of this green RNA, because the cell is going to make a whole bunch of copies of it. This way it just looks like you're getting one for one. There'd really be thousands of these that are transcribed in this to make the virion. So that's an example of a positive stranded virus. Let's look at an example of that happening in the cell. So this figure, I think with this figure they're giving us the example of poliovirus. Let me make sure that's true. 2724.
Yep.
Is that right?
Yep.
Poliovirus? Thanks. So this is showing poliovirus. We talked about how it gets in before. It binds to a receptor, maybe on your gut, maybe on your nervous system. Poliovirus can be found in water. When my mom was a kid, back in like the 1950s, she wasn't allowed to go swimming in the lake because they were afraid she'd get polio, which sounds ridiculous to us, but that was before everybody was vaccinated. And polio was shed by, was it fed by birds? Was it ducks too? Or maybe it was just that all their septic tanks drained into the lake and they were worried about it. It's just a human thing, isn't it? So they're worried about anyway, long story. The reason they're worried about getting it out of the water is because you drink the water, it's set up shop in your digestive tract and multiply and then get into your nervous system. So here it is coming into the cell. Positive strand gets out. It makes the negative strand for genome replication, many, many copies of this, and it makes what's called a polyprotein. So here's something different about the virus's proteins that are important. Because their genome is so streamlined, they don't have space between their genes. They don't initiate transcription in a whole bunch of different places or even have space between them like bacteria do with their operons. They make one big protein and then they process that up into several smaller parts. So this is showing a polyprotein that gets made, and then it goes through proteolysis to make individual proteins that need to get brought along and packaged into here. So these individual proteins include the capsid proteins. What else would be super important for that? What other proteins would be in there? It'd be the protein that does this job, right? I'm going to write that down in a second. They get packaged and it gets out. So what happens with replication of all viruses, with their RNA viruses, they use an enzyme called RNA-dependent RNA polymerase. And there's two kinds of activity that RNA-dependent RNA polymerases can have. One of them is called the transcriptase activity. It's the one that's generating the transcript, which is what? It's the mRNA, right? So this converts the genomic RNA into mRNA. So this genomic RNA can be negative stranded potentially. The other activity is known as viral replicase. So the replicase function is taking the single-stranded RNA genome and converting it into the double-stranded RNA genome. I shouldn't say genome, double-stranded RNA. So we'll look at some examples for that in a little bit. Let's look at, in fact, first of all, do you guys have any questions about this before we go on? Because this is the one that seems a little more conceptually, Ben.
So if it was a positive RNA genome, would it need transcriptase activity? Because that's already functioning as mRNA.
Sometimes they're together, right? Because that would be like this one. So in this case, it's making, so the reason that it is, what would make this figure more clear is if it showed that this happens many times in order to make enough protein, right? So it does have to make this, which is used as transcript and as genome.
Okay, so
So they get combined.
Can you just, are mRNA and positive stranded RNA just interchangeable in all these scenarios?
You don't call it mRNA when it's the gene. So it depends on what the function is, unfortunately.
Okay.
I mean, the molecule is the same thing, but when it's used for a different function, you would refer to it as the genome.
Okay.
So if it, as it's getting, when it goes this way, you're thinking of it as the genome because it's going to be going into here. When it's going this way, it's being used as the transcript as mRNA. Yeah. Tony.
Say you're referring to like the positive strand RNA just as the genome.
Yeah.
If it's used as the genome, will it ever also be used as mRNA?
It does get used as mRNA as it comes into the cell. Because as it comes into the cell, it's available for transcription, or for translation right away, right here. The genome gets used as mRNA, yeah. Right, Luke, were you gonna have something?
Yeah,
Somebody else?
does he use the host and translation machinery then?
It uses the host translational machinery. Sometimes viruses have a tRNA or something associated with them, but they have to use the host ribosomes. and most of the tRNAs, and all the amino acids that the host made and everything. Yep. Okay, let's see here. So we talked about that. Here is the example for what happens with the negative stranded RNAs. And this shows a little bit more detail about the processing and about the spike protein formation. So let's look at this again one more time before we do this. The RNA comes in as a negative strand. It gets transcribed into positive RNA by the RNA-dependent RNA polymerase, and it's used both as a transcript to make protein and as a template to make the negative strand. So this would be the viral replicase function. It makes negative strand, which is then used to make the genome, right? So that's what happens with influenza virus. So here's influenza virus. Same thing, it's got the spikes, it binds here to some sialic acid residues for influenza, we'll talk about that later. Gets in through the coated vesicle. The RNA, which is the genome, gets into the leaves the nucleocapsid down here, gets to the nucleus, and then it has to go through to make positive single-stranded RNA for two reasons. One is so that they have a transcript for making the proteins, and one is so they have a template for making the genome, right? So there's a bunch of detail in there we're not going to go into because we've got to be realistic here. Let's be realistic. So some of these transcripts get used to make the capsid, and some get used to make the spikes. Here we're showing the transcript that gets used for making the spikes. Coming down here to the rough ER, that's the ribosome. And these proteins get processed and put into the membrane of the rough ER, and they go through the Golgi down here. And in this case, they get inserted to the plasma membrane of the host, and the virion gets formed as the nucleocapsid comes down here and buds out of the host. It's budding off the surface of the host and taking the membrane with it. So the host has to be healthy enough, because what does it do? All of this requires activity of the host, including the cytoskeleton, processing, the translation. All of that stuff has to be going on in a healthy way in order for the virions to get out of there, right? So you can't just kill the whole entire cell all at once and have everything work functionally in order for that to get out. Some kinds of viruses kill when we talk about the bacteriophage, we'll talk about how they kill the whole thing all at once and lyse the whole cell open, but they don't get an envelope when they're doing that. So there's some cool stuff that's going on with these proteins, but like I said the other day, we could take a whole entire semester to talk about viruses, or maybe a whole year, but we got to cut it off somewhere, so you're not going to be responsible for that, Ben?
So in general, the translation is provided by the host.
Yep.
The transcription is provided by the virus.
Yep, that's right. Because of the fact that their genomes, so their genomes are what are diverse and weird, right? They still use the same amino acids. Well, there's a couple of weird exceptions to that rule, but in general, they're using the same amino acids. So we don't have time to talk about retroviruses today. We're going to talk about them next time. If you guys want to read, so next time, the reading assignment for Chapter 3, the pages we're going to cover for Chapter 3 are on Moodle. We're also going to talk about retroviruses and what happens with retroviruses in terms of replication. Finish a little bit about viruses and then start talking about bacterial cell biology. Does anybody have any questions? We have 5 perfectly good minutes, but I'm not going to start retroviruses with only 5 minutes to go. Hannah, you're presenting next time. Salmonella? Questions, anybody? All right. See you Friday.
I'll still have to read retroviruses. Because I got through herpes and then all my rants. What now? You have biochemistry? Shucks, I can't go to campus nurse right now because I'm going to be on like a lunch break or something. It's noon.